can be covered by fire, right? Even ability to evolve, because it depending on the surrounding, how much oxygen you have, what are the material, and most of the capacity for self-replication and self-assembly. Like even mechanism for sensing and responding alteration in surrounding, most of the point can be covered by fire also, right? But this is just, just some interesting point. That's why I told. But whatever I discuss about living organism, these are the major points should be there. So as a result of that, let's see, in today's world, like there is one term that is called biomimetic. That means, since as just I explained that living system is very complex and nicely organized and assembled thing. So the idea is if you can learn something from living system itself, because whatever you can see today, that is the result of years and years after evolution. So our goal is to learn the living system to some extent, and if possible, try to gain some knowledge from living system and imply that and try to use that in our own way, right? So that is another field that is called biomimetic. So design something, make something, seeing some living example. Okay. Just a minute. I will. Okay, so somebody already started recording. That's good. Um, just okay. Just I recorded the attendance. This is, just to check how many students are attending classes. So here, I will give you some example, um, like which are actually like very important discovery, or I would say those are very commercially successful discovery, but that is based on some living system, like based on our example, they like scientists, they got inspiration from living system. So here you can see this plant. This is some kind of cocklebar. So many of you have seen this plant. They have like, you can see those fruit, and if those fruits are dried and if it stick to your hair and trousers, it is very difficult to take it away. Okay. So now the point is why I'm showing this. So this is example of some kind of bio-inspired discovery, and this is one of the most successful one. So just one story I would like to tell you. So here I'm just showing that if this fruit actually, just stuck to your socks or somewhere else, then it is very difficult to take it away. But what happened long time back in 1941? Okay, so one electrical like engineer in Switzerland. Okay, so he was just walking in woods in kind of forest land, and some of those fruit that actually stuck to his trousers as well as his dog, like dog hair. And when he came back, so he was trying to, just take those things away, but he cannot. So it is kind of very tightly sticking together. Then what he did, so although he was an electrical engineer, but he got very much interest on that. So he was studying under microscope how those small seeds or like dried fruits, they are stick together like with trousers. And then he actually found that in trousers you have like small like loop of cotton or whatever the material, right, the trousers made up of. And in those dried fruit, you have small hook-like structure. So he actually tried, tried to understand that for many years, and then finally he come out with the solution that you can make some kind of material which can stick together, and that is you are using today, like all of you are using today, even right now many of are using. So that is the discovery of Velcro. Okay, all of you, like your shoe, your bag, everywhere you use Velcro. Velcro is commercial name. So Velcro actually came from this word, I think French word. So velour. So this is that means velvet, and crow from crochet. So that is some kind of hook. So what he found that uh, in case of this fruits, they have small hook-like structure. So then actually George de Mestral, that scientist, that engineer, so he actually designed this material, and finally commercially this is one of the most successful discovery, I would say, that throughout the world people use this Velcro, and now the modified version with better material, right? But this is came from this story actually, whatever he faced a long time back in 1941. Next, here you can actually argue with me, like, today I will not discuss too much about DNA, but just, just discuss this, some of this point. Uh, then you can argue with me. You can ask me actually that, sir, why I need to learn biology? Because this is what he has done. That is nothing but good observation. You are absolutely right. Right, you are right actually, that this is nothing with, like, something biology. Just he was very much, like, observing those things very critically, and then he invented that. Yes, you are right. So let's see another example. We will go, like, we will discuss few more examples, then you will understand why we need to know biology also. So you can see that this is a bullet train. So now bullet train is available in many countries, right? But mostly bullet train actually initially like started running in Japan. So what happened in Japan? There are a lot of tunnels in their railway system, and when those trains, they are actually entering inside the tunnel, right? And they are going through the tunnel, and they are emerging from the tunnel. Just at the front of the engine, there is some air pressure built because it's going through tunnel. And whenever at the end of the tunnel, that air pressure get released, and it creates a lot of, like, big sound, what can be heard from long distance. And also you have to remember that Japanese, they are always trying to, make their train faster and faster. So that is again another reason why the sound is more, and that's create a lot of problem in sound pollution, and it's really creating problem. So they are actually trying to come up with some solution. And during that time, what happened? Another railway engineer, his name is Izzy Nakatsu. So you don't need to remember nothing this thing. Just, I'm telling some of the stories, and I will tell you what to remember. Okay. So Izzy Nakatsu, so his hobby was bird watching, right? So he was working in some Japanese rail company, and he observed that some fish, say all of you know this fish, this is kingfisher. So when it actually searches for meal, that means that it want to catch some fish, it will not create any, like, disturbance or any ripple in water. It just goes straight away, and almost all the time, like almost like 99% like, success that catch the fish without creating any problem. So then this scientist, like this engineer Nakatsu, so he was, like, trying to design some kind of, rail engine, the front part, I would say. So he just replaced that with the kingfisher head. I'm like telling the model, not like kingfisher head. So this is almost 50 feet long, like still in like still like material, and you can see this is very close to, or it's resemble to Kingfisher Head. And as a result of that, it solved the problem of that, that whatever the air pressure building in front of the train while it is going through tunnel. Not only that, it helps, or I would say it increases fuel efficiency also. So as a result of that, all the way, it is very much beneficial. And now these kind of bullet trains are available in many places, and it can run very fast. In Japan, it can, like, in conventional tracks, railway track, it can go above 300 kilometer per hour, and in magnetic levitation system, right? So I believe that most of you know what is magnetic levitation system. Through that system, it can go even more than 600 kilometer per hour. So you can understand, right, how speed is. So 600 kilometer per hour. So in this case also, you can still tell that this is again another example of good observation and its, execution. But in the next one, you will see you need to understand biology, like not to some extent, in a great extent. So here, this is like small organism, small living thing. So this is, I would say, long distant cousin of some kind of arthropods, some kind of cockroach. Okay, so its name is Tardigrades. Okay, so forget this name. You don't need to remember this name. So this is a small organism, like, some kind of cockroach, but very small, millimeter long. So what's the importance of this thing is, so this organism, just like seed, even better than that. So it can stay as almost like non-living material for 100 years. So although this is an animal, but in unfavorable condition, it can stay there for 100 years, more than 100 years. And even in high temperature also, you don't need to refrigerate it, right? Even it can stay more than 100-degree Fahrenheit temperature for more than 100 years. So you can see, so that means some mechanisms are there so that it can still survive after 100 years. So this is very unusual if you think that this is an animal, right? Now scientists try to actually understand what is happening inside this small creature. So they found that they have some mechanism so that they can store their biological molecules which are important for their life. Say, which are the biological molecules? Tell me, what are the major biological molecules which are very much important for our life? Can you tell me, just one student? Just unmute yourself and tell me. That biological molecules which are, yeah, very good. Protein, nucleic acids, right? Carbohydrates, those are very important macromolecules which are absolutely, yeah, mostly I would say nucleic acid and protein, and also carbohydrate, definitely. And now I would say scientists try to understand how this organism can stay for that long, like, in unfavorable condition. And if you can learn that, then you can use that for our own good. I will give you one example that, for example, like vaccine, like many poor country, they cannot vaccinate properly to their kids, not like this COVID vaccine I'm saying, like small kids, they are taking a lot of vaccines. But the major problem is the transportation, in particularly in poor country, and like refrigeration. So you have to store your vaccine, which will be, provided to all kids when they require. But you have to store it properly. But in many poor countries, like, like I would say really poor country, so there the refrigeration system is not good, the transportation system is not good. So as a result of that, many of those vaccines actually they get lost during transportation or storage. So if we can learn this system, so we want to keep our vaccine also in such a way that it can stay in room temperature for a long time, years after years. and even it can withstand, like high temperature as well as for long time. So two company, one is Biometrica, one US-based company, and another company, Nova Laboratories in England. So both of them, they actually understand this process, and they made some kind of, I would say, procedure so that they can keep vaccine in certain material so that it cannot be, spoiled in high temperature, right? So this is called candy coated vaccine. So they like, they made some kind of material, and so that it can preserve those protein, like, this animal they can preserve, and they can keep their protein nucleic acid intact over the long time. That way also scientists improve this thing, right? So in order to achieve this, you need to understand biology and chemistry to some extent, like otherwise you cannot do. This is not just based on observation, right? Now, next is even more interesting for you guys, the current data use. So every minute you can see that how much data is being used throughout the world. This is, I have taken from Google in 2020, like, last December. So the thing is, the problem, what I want to say, the data storage, where you will storage, right? So now actually another important field is coming up, like that is called digital DNA. Is anyone of you actually heard about it? If you know about what is digital DNA, you can unmute and you can tell me. So in class, I asked actually several questions. Okay, so if you tell wrong answer, nothing will happen. This is just my normal practice so that you will be in loop, right? So we can discuss. So I can also learn some of those things from you, and you can learn some of those things from me. Okay. So as a result of that, normally when I ask question, try to answer. Okay. And if you don't know, you can tell, okay, I don't know. That is also fine. Nothing will happen, but it's good if we discuss together, and then we can try to understand and learn something. So DNA,
So I guess
yeah,
digital it's just the
yeah.
code of like the TAGCs, whatever. Like during the COVID, like COVID discovery, like codes of the genetic codes were sent throughout the world for the vaccine purposes and research purposes. So I guess that's what digital DNA.
Okay, okay, Sama. So thank you. So I'm just, like very briefly tell this, and then if you are more and more interested, you can go to some literature. I will send you one relevant article also. So digital DNA, the first thing in the email yesterday I mentioned you that what's the function of DNA? What it does? DNA store information, and whatever is happening in our body, like those information actually written in DNA, the genetic information. So it can store information, lot of information. You cannot imagine actually how much information in the terms of some nucleotide sequence that I will discuss like after a few minutes. Some nucleotide sequence like ATGC. So whatever some have tried, like, some kind of code is. So what happened in 2015, sorry, not 19, 2015, Microsoft, they started like, one project that is called digital DNA, the future storage device for world's data, and some other like, company and other academic institute, they are also trying this. So then what are the advantage and disadvantage to store information in DNA? First, like get it clear that DNA present in our body, like our every cell, like we have in an adult human being, we have approximately 10 to the power 13 cell, 10 to the power 13 cells. Each cell, they contain DNA, huge amount of information. I will discuss more details that how many nucleotides, how many, how big this DNA is later, right? But the thing is, all those cells, they contain this DNA and that contain information. And that information gets processed rightly so that you are healthy enough. Okay. So everything is going in a right way. That's why we are healthy, right? Everything is happening rightly. So the idea is here that if we can store information, like instead of binary code, like all of you know, like instead of 1 and 0, if you can use this system like ATGC, like you are getting some advantage. So, and if you can store information, so see some of those like drawback or advantage of DNA compared to hard disk and flash memory. So if you see the read and write speed, that is actually one of the drawback in case of bacterial DNA. So any DNA, forget about bacterial DNA. The thing is you have to write the information in DNA if you want to use as storage device, right? So this read and write speed is actually little bit difficult, at least today. Maybe after 50 years, 100 years, I don't know what is going to happen. But in hard disk and flash memory, like I'm not spending a lot of time here, but all of you know how fast you can read and write. Now, one of the most like advantage is data retention. So the data retention in terms of years is more and more in case of DNA. You just put information in DNA, like you just code and you can just store information in DNA and then keep that DNA in refrigerator. Like if you are keeping it, I would say in minus 20-degree temperature, or I would say minus 70-degree refrigerator, it will stay for years after years, not just 100 years, 1,000 years, right? All of you know, even some from some fossil, right? Like fossilized thing, you can still extract DNA and still you can, you can extract some of those information. So as a result of that, if you can store DNA in right condition in refrigerator, you can store your information for long time. But the challenge is, as I told you, the first point here, read and write speed. Power usage, data density, all are good in this case. In case of DNA, I will send you this article. This is only 3 to 4 pages article, came out in Nature a few years back. So if you are interested, you can go through it. But this is not for your exam, just for your interest. Okay. So now here I told you that Microsoft Corporation, they started some project DNA storage research, and you can see here, I'm not going through it. In that article you will find it, what I will send you, that they tried to quote one poem from Shakespeare's book, and there they coded in DNA. Actually, that information has been coded in DNA itself, and I'm sure that some more advancement already already there. I just made this slide few years back, but if you are interested, I'm sure that you will get some more information. So that way you can understand where I started, that some cockle bar that stuck to that Swiss electrical engineer Stauser, and then some vaccine, storage of some vaccine, then how you can store information in DNA. So the field is very wide actually. Just you have to take the advantage out of it. So also if you see, like any good engineering institute throughout the world, even like whatever all the IIT system in India or abroad, whatever you think that it is good, you will see now in their undergraduate level they have biology course. Primarily because of we don't want that you have to remember a lot of things. No, if you see many of those like engineers when they are growing up and they are joining in academics or in company, many of them they are dealing with some biological problem, because either biological problem or they are trying to get some solution from biology. So this is very common. Whatever department you have joined, some of you, if you are in mechanical engineering in IIT Kharagpur, some of you computer science, electrical engineering, all department, like I know that all students are here, so but I'm not telling all of them, but you will see almost every department, at least 1, 2, 3 faculty members, they are working with some biology related problem, because this is now common trend, and the field is more wide, and that I would say the scope is more wide also, right? So uh, here, this is the last slide. So here, one interesting fact I would like to discuss. His name is William Cohen. So he is there still, like in Carnegie Mellon University. So in machine learning department. So what happened? Almost 25 to 30 years he taught about machine learning and mostly like, artificial intelligence and all those things. But he actually observed that many of his peers, many of his colleagues, they started learning biology. They are trying to contribute in biology. But one of the major problem was that the biology books, most of the biology books, they are written in such a way, it is very difficult for engineers who know they don't have biology background. So as a result of that, it is very difficult to understand what is written there. That is the communication gap, right? That some like, from the very beginning, from the first chapter, it started with lot of jargon, lot of nomenclature, and it's a problem. So what he did, although his background is machine learning and artificial intelligence, but he wrote one biology book. Like you can see here, is the A Computer Scientist Guide to Cell Biology. I downloaded this book and I read this book so that I can understand some of the problem engineer they actually face when they are going through biological subject. So also like the course is designed in such a way that you will not face that much of problem. At the beginning, I told you that your responsibility is to attend the class and patiently try to understand that what we are discussing, because I know because some of you are like right now facing some problem, connection issues, all those things. But just try to concentrate, and if you have any problem, write me email and discuss with me. Okay. So now actually from this point, I will actually go into the details of I would say DNA, right? Whatever topic we are going to discuss. So if you have any question, any query, like not what about I discuss right now, any like general query, you can ask me, like 2-3 minutes, like break. So you can ask me, and then I will go to the next topic, like actual topic. So what about the internet connection? Is it okay? So you can understand, right, what we are discussing?
Yes, sir.
Okay, very good.
Yes.
Yes, sir.
Yeah. So see, I would like to tell you that I understand also like students' point of view. This is very difficult for online classes, like the full semester, all the subject you attend, like online classes. So from like from 8 a.m. you started every day, you have like many classes, like I would say many hours just in front of computer or mobile phone. This is really problematic, I understand. So that way, I would say the course is designed not that many, not lot of topics included here. Mostly like some of those interesting and important topic which you need to know, and that will be better. Maybe that is not directly related to your core subject, like whatever engineering field you join. If you like, if you are, but if you are learning this thing, like if you take some interest, then it will help you at some point of your life, like to some extent, I would say. At least you will know some of the important fact about biology, which you need to know as a normal, human being. That is another important thing, right? So let's discuss about our components of living system. So all life forms on Earth are composed of building blocks that are made of combination of carbon and other elements, right? So carbon is the most, like, carbon should be there, and then hydrogen, oxygen, nitrogen, sulfur, phosphorus. So those, like, atoms are present. So for example, protein. So they have like many of these element. Apart from phosphorus, in protein you have, all other things here. So that way, all these elements are very much important, and then they finally make some kind of macromolecules, either that can be proteins, carbohydrates, lipids, or nucleic acid, right? Those are the important things. And in first two classes, we will concentrate on nucleic acids, and then we will slowly go towards protein, that how, so here I should mention here that many of you, like you might know during your school days, that information is stored in DNA, but DNA itself, it cannot work. Only it's stored in the information. But what is the working thing? Like who can work? Proteins, right? Proteins present in our body, they can work. So sometimes I say that whatever present in our body, either that is a protein or that is a result of a protein, right? So proteins are the functional molecule present in our body. But how you will get that protein, right? You need some information in order to make the protein. That information is written in DNA. And in the middle, you have some process, some mechanism, some steps that can decode the information. What information? The information which is written in DNA. And after that decoding process, you will get the protein which is functional molecule. You are eating some, some food like rice, bread, whatever it's there. It needs to be digested. It will be digested by some enzymes. Enzymes are proteins, right? Enzymes are proteins. Those are the functional molecule. And the information to make those enzymes or proteins, it is written in DNA. And I told you the decoding process, that is called transcription and translation. Whatever, I will send you the whole topic, the syllabus, you will get that one topic is transcription and translation. That is the decoding process, how you will get this functional molecule out of DNA, right? So now, if you see here, like in this case, like first two classes, I will concentrate on nucleic acids, then how the information gets processed so that finally it will make some functional molecule and it will function in our body. Without functional protein, we cannot survive for a second. And then the genetic engineering and its impact on society. This topic can go to the last at the end, but intentionally this semester I kept it here because whatever we will discuss here, like the prerequisite will be taught in these classes. So as a result of that, it will be good if I discuss here. And because in short time period we are actually finishing 2 semesters, that's why only we have a few classes, right? Only we have 9-10 classes. So as a result of that, right, we actually opt out some of those like topic we are not discussing during this semester, but it's okay. So after that, whenever you learn about these biological macromolecules nicely, then we will discuss about what is cell, what is living system, how those macromolecules are coordinated, how they're functional inside the cell, and how they make living organism, right? The growth and development, and particularly stem cells and its biomedical potential. Then we will discuss about how our how our body defend ourselves, right? That is called host immune system. But before that, we will discuss virus and human diseases, that why we need to defend ourselves, right? There are many or various challenges that we are facing constantly. So that's why we have to defend. So that way the course is designed, and we will discuss and we will go through it just step by step. And I hope that most of you will enjoy if you just concentrate on the class. Here, nucleic acid. The nucleic acid here, I will mostly discuss in this perspective that how is genetic information stored and maintained, right? So initially, I will actually discuss one experiment, like that, a very important experiment. I will discuss that, and then we will go into the details of DNA. So as I told you that our intention is not to provide you a lot of information. So let's discuss about the experiment first. The experiment that proves DNA is our genetic material, right? So I will use my whiteboard so that I can go through it properly and you will understand also, right? 1 minute. So here, uh, this is called the Griffith experiment. Griffith is scientist name. Okay. So remember this name actually. I told you forget everything, but this one I'm requesting you to remember this Griffith experiment in 1928. You don't need to remember the year, but why I told you to remember Griffith experiment, you will understand. This is very important experiment in biology, particularly in modern biology. So the goal of this experiment is to explain that DNA is our genetic material, right? All of you know now that DNA is our genetic material. If you go back in your school days, in class 8, you read that Mendel experiment, that Mendel, he was using some, some plant and some genetic experiment. So the field of genetics was known during that time, right? But why those informations are present, that was not known. Like he crossed different types of pea plants, and finally he came out with some theory and which are actually true. But the thing is, how those informations are stored, where it's stored, what are the material, the chemical properties, nothing is known. So Griffith's experiment is the first experiment that indicate that DNA is our genetic material. The information is stored in DNA. This is the first experiment. Although he was unable to finish the experiment, right, completely, because actually he passed away. While he was doing experiment, he contributed, and he mentioned his outcome or result. But the problem was, he cannot finish because in 1941, while he was working in lab, so 1941 means, you understand, like that is during time of Second World War. So because of some German bomb actually hit his lab, and he also passed away. He didn't get Nobel Prize because after death people, they don't get Nobel Prize. But his contribution is huge. Okay, that actually I would say started understanding that DNA as genetic material. Let's discuss that experiment first. Very simple experiment, but I will go through it step by step so that you can understand. Griffith, he was actually trying to make some kind of vaccine, and DNA is genetic material. That discovery is I would say, some kind of accidental discovery. If you know that most of the big discoveries, those are actually accidental discovery. They are doing something else, and during that time they've observed something completely different or very important thing. So what Griffith was doing to make vaccine against some bacterial pneumonia. So what happened in 1918, one pandemic happened. Have you heard about what is this pandemic? Now you are actually sitting at home because of pandemic. So do you know what is the greatest pandemic? Like what,
So Spanish flu,
Excellent. So
influenza,
Spanish flu.
and
excellent. Very good. Yeah. So in 1918, it was Spanish flu year, right? During that time, like almost, I would say, even you can search in Wikipedia, like some basic information, you will get it there. So like million people actually died. Like you can see that time world population is not that high, right? Almost, like reports say, around 5% of world population died because of Spanish flu, some kind of viral flu. But you know what happened during flu, like if like people, they are very much sick because of viral flu, their immune system is not very in a good condition. What happened? There are lot of bacteria which are opportunistic bacteria. That means those bacteria in general, they will not affect you. In general, that will not create any harm to you. But when you are actually sick, you are weak because of some viral infection, those opportunistic bacteria, they will again infect you, and they will create more and more problem, right? So as a result of that, the people died during Spanish flu, like almost 5% of world population. as I told you, those are not direct result of viral flu. Rather, this is secondary infection. That means people are weak that time, and some bacterial infection. The mostly I would say the important bacteria that causes this problem is Streptococcus. Again, if you forget this name, nothing will happen. This is bacteria. Streptococcus pneumoniae. Okay. So here, this is the bacteria, and many scientists across the world, they were trying to make vaccine or some kind of some remedy against this bacteria so that they can save life, right? So Griffith was also doing this, but while doing this, he suddenly discovered something else. So let me discuss his experiment. So there are two types of Streptococcus pneumoniae available during that time. Okay. So any one of you like aware about this experiment? Sometime I asked this and 2-3 students, they told yes, I know. So if you, any one of you know this experiment, tell me quickly.
Yes, sir.
Okay. So most likely you have you had biology in +2, right?
No, sir. I mean, I studied for ICER exam, so
Okay. Okay. Very good. Very good. Yeah, I believe even 10+2 it is not covered. So anyway, so but this experiment is very simple. Let me go through it. Thank you. So Streptococcus pneumoniae, there are two strains. One is called R strain. R means their surface is rough. Okay, this is bacteria. Their surface is rough. Okay, this is called R strain. And the other strain is smooth strain. S strain. So now I will not say rough and smooth, I will say R and S. So this is smooth strain of bacteria. Two different strain. Now the thing is, this strain is called virulent. This term might be unknown to you. Virulent means, I would say, that causes disease, that causes problem. And this strain is non-virulent. That means it will not create problem or it will not cause the disease. Non-virulent, right? So Streptococcus pneumoniae, two different strain, and one is non-virulent, not cause the disease and it will not be harmful, right? But the smooth strain is virulent. Why? Because the problem is, when this bacteria infect us in human, if you imagine, so we have our own immune system. So that will try to clear this pathogen, that will try to stop this bacteria to grow. Now the thing is, because of this smooth strain, so they have on the wall, on the surface, they have some polysaccharide, okay, polysaccharide layer. That's why this bacteria is smooth, and that actually, escape, that help to escape our immune system. So our immune system cannot handle this bacteria, smooth bacteria properly. So as a result of that, that can cause disease in our body. But non-virulent, the rough one, our immune system can handle it properly, and as a result of that, it cannot cause the disease. This is what I told in context to human individual. But while you are doing the experiment, when Griffith did the experiment, you cannot, use human being itself in order to do the experiment, right? So what he did, he used mouse. That is normal, right? He used mouse to do this experiment. What he did, so he actually, take a mouse, okay, healthy mouse, and now he inoculated. Inoculated means he injected R strain of bacteria, the rough strain of bacteria. This is live, okay, living bacteria, not dead. So then what will happen? Tell me quickly. The mouse will die or mouse will survive?
Survive.
Very good.
Survive.
Yeah, because
Survive.
the rough strain is non-virulent or avirulent. So mouse will be happy, no problem. Okay. It is right now fine. Now, what happened if you A strain oh sorry, I should not write here. So now in this experiment, okay, so in this experiment injected smooth strain of bacteria, then what will happen? Mouse will
Die.
yes die, because the smooth strain is infectious, it's virulent strain, the mouse will die. Now the next experiment is also, let me explain, and you should try to answer. Only 4 experiments he did, and then came out some solution, good solution. So here what he did, this is also live S. Here what he did, heat killed S bacteria. Heat kill means, you can just boil that bacteria, like some kind of pressure cooker, right? Autoclave, like you can just sterilize, you can just boil this bacteria. Then what will happen? What you understand? Like you expect that this mice will survive or die?
Survive.
Yeah, very good.
Survive.
Survive. Survive.
Survive. Very happy because the bacteria is already dead. Nothing will happen. So these are very straightforward, I would say. Whatever I explained here, like rough and smooth strain, based on that you can provide a right answer. The last experiment was the most important one. So what he did, he took this mouse and he mixed 2 things here together and injected. One is live R strain plus heat killed A strain. Then what will happen? Tell me, whatever you understand, right? What might happen?
Survive. Survive.
Survive. Die.
Survive.
Yeah,
Die.
so I expected this mixed answer, because if you say it will die, that is actually, I would say theoretically that is correct, because whatever until now, whatever you will say that mouse will survive, because the R strain cannot cause the disease and the A strain is already dead because it's autoclaved, it's heat killed. So the answer will be that mouse will survive. But surprisingly not, that is not the case. Actually, mouse will die. Mouse will die. This is, I would say, during that time, this is very, very interesting thing, interesting result. Mouse will die. Because if you understand all these 3 previous experiments, then apparently it seems like, the mouse will survive, but mouse died. He repeatedly did this experiment again and again. Every time mouse died. Right? Why? Because then the theory is coming here that something is happening so that the heat killed S strain, the heat killed smooth strain, that bacteria is dead, but some of those material present inside the bacteria, that is still active, the chemical material, they are still active. When you are mixing together this dead A strain of bacteria, heat killed A strain of bacteria and live R cell, somehow those live R cell, live or the rough strain bacteria, they get transformed into smooth strain, and then that can cause the disease. This is one of the very important and surprising discovery. So this is called transforming principle. That means Griffith actually cannot explain all those things. As I told you, he passed away after a few years. So what happened? Later on, some other scientists, they actually proved that what Griffith did, that was absolutely right, that when he mixed this live R cells and this heat-killed smooth strain, some information present in those heat-killed cell, in those smooth strain, that actually transform those R cell into the S cell, those avirulent bacteria into virulent bacteria. So as a result of that, that can cause the disease. So did you get my point?
Yes,
So,
sir.
yeah, okay.
Yes, sir.
Yeah, so this is called transformation or transforming principle. This is very important in modern biology. Like almost every day we use in our lab, we were working with genetic engineering, protein engineering. So my lab particularly, we do genetic engineering, protein engineering in a routine basis almost every day. So in this lab, this principle, like we like regularly carry out this transformation in a defined way. In a, I would say, this is just discovery how it has been discovered, but now we can experimentally we can do it very quickly. Later on in the genetic engineering class, I will discuss about that. So here, this is the experiment, and the idea is then whatever I told that although this smooth strain bacteria, like they are died already, but they are like, they are not live anymore, but the gene or the DNA of those bacteria, that DNA can transform those rough strain bacteria so that that can cause disease. But who proved this? Few more scientists later on, whatever Griffith did, they were continuing this thing. Quickly, I will go through it, and here, like nicely mentioned, so I will just go through it and try to understand. So this is, so now inside the, as I told you that here you can ask me one question, that that whatever experiment I told, based on that I can only tell that this dead smooth bacteria can transform R cell or rough strain bacteria. But who told me that this is DNA? This is not protein. This is not carbohydrate, not lipid, right? Because I just know this is DNA, that's why I'm telling you DNA. But based on the experiment, you shouldn't know yet. You can only, like whatever I'm telling you, that this bacteria transform R bacteria into S bacteria, smooth bacteria, so that it can be lethal. It can cause the disease. But what, like, why I will say that DNA is the genetic material, it contains the information and DNA transform this thing. So that answer you will get in this slide. So what they did, this heat killed S cells, that smooth bacteria, they actually, like, they grind those bacteria, they make some kind of solution. I'm not going into, like every minute details, but from those solution, they extracted or they removed lipid and sugar. That is possible. This is based on some biochemical experiment, biochemistry. So they remove lipid and sugar. Then whatever solution present after removing lipid and sugar, that means sugar means here carbohydrate, okay? So that solution they distributed in 3 different tubes, and in one tube they add proteinase. So in biology, particularly, like all enzymes, they end with AAC, proteinase. So what is the function of proteinase? Proteinase will break or digest protein. Okay, so no protein here. So in solution, all protein will be degraded, will be destroyed. Why? We added proteinase. In this tube, we add RNase. From the name itself, you can tell AAC for enzymes. RNase means we have nucleic acid, 2 types of nucleic acid, DNA and RNA. So if you add RNase in this solution, no more RNA. Okay, RNA will be destroyed. Then in the third tube, you added DNase. Similarly, DNase will cleave, will destroy DNA. So no DNA in this tube. And then what happened? They take sample from this tube, the second tube and third tube. So in this conical flask, and they added live R cells, rough strain bacteria. So as a result of that, later on they did experiment and they found, okay, S cell already appear out of this R cell. So that means from this experiment you can tell that protein is not involved in this transformation. Why? Because you already destroyed the protein. Protein is not there. So protein is not responsible here to make those R cell into S cell. In the second experiment, when they added RNAs, right? No RNA, they added R cell, again S cell appeared. So you can tell, again, RNA is not involved here. No RNA, still S cell appear. So let us discuss about the third experiment. When they added DNAs that destroy DNA present in the solution, and then in that solution, they added the rough cell bacteria, the rough strain, then no S cell appear. That means they depend on DNA. You need to have the DNA present in the solution in order to transform this R cell into S cell. So now we can say that DNA is our genetic material, or I would say DNA is genetic material, not our, because it's in bacteria, but later on, we found that in every living organism, DNA is genetic material. Okay, so I hope that all of you understood this experiment if you are here in the class, right? It's clear, right?
Yes.
Very good.
Yes, sir.
So
Yes.
yeah, so take 3 to 4 minutes break, okay, and then we will again start discussing. Every class I will give you like few minute break, so you can like 3 to 4 minute, 5 minutes break, and then we will discuss again. One more thing. So do you have class after 10? Most likely you don't have class after 10, right? The 10 to 11 is
No,
free
sir.
slot.
Yes, sir.
Yeah. So normally what I do, I try to go very slowly so that you don't need to, as you can, like by this time you understood. I will even tell you, even you don't need to understand, like, read later. Just try to attend the class properly. Just go through the slide one more time. That's all. You don't need to know anything else. Like if you are interested, just read so many things as you wish, but attend the classes and try to understand. And normally I extend the class like 10-15 minutes extra just so that I can go slowly and I can understand and I can explain whatever is required. So as a result of that, please take break for 3 to 5 minutes and then I will start discussing the next part. Okay.
Okay. Okay.
Yeah, thank you.
Excuse me, sir, I had one doubt.
Yes, sure.
Sir, when we mixed R and